Mushroom research advances constantly, and the great discoveries may be around the corner. Unfortunately, life in a mushroom farm is always busy, and it's hard to stay up to date. Specialized scientific journals don't help due to technical language and frequent paywalls. Our mission is to bring the latest discoveries on mushroom science to the industry professionals and the mushroom people. Ladies and gentlemen, please welcome to the new episode of the Mycelium Digest.
Welcome along to the Mycelium Digest. This is episode 2. Today we're doing a deep dive into a compound you might not think much about, but it's surprisingly important: calcium oxalate.
It really is fascinating stuff.
Our mission today is to sort of unpack what it is, look at how tiny little microbes interact with it, and then really zeroes in on its role in growing white button mushrooms, Agaricus bisporus. Specifically, how it might affect, the yield.
Yeah, there's some really cool science there. We've pulled together insights from research papers, some amazing electron microscope images, and microbial studies.
Should be a good one. Just before we jump in, a quick ask. If you're enjoying the Mycelium Digest, we'd love your support. You can subscribe on YouTube, maybe hit the like button, or leave us a rating on your podcast app. It honestly makes a big difference.
It really does. Okay, calcium oxalate.
Right. So what exactly is it and where do we find it?
Well, fundamentally, it's a chemical compound, calcium bonded with oxalate. But the really striking thing is how incredibly widespread it is. It's a major secondary product, meaning organisms don't need it for basic life, but it serves other specialized roles.
Okay.
And we find it everywhere. Plants, animals, fungi, bacteria, they all can produce it.
So it's not some rare, obscure molecule then?
Not at all. Get this, it's found in over 215 families of flowering plants and conifers, and in wood of over 1,000 different tree genera.
1,000? Wow.
Yeah, and in some plants it can be like over half their dry weight.
50%, just calcium oxalate. That's huge.
It's an enormous amount, so it's clearly playing some significant roles.
Okay, so what does it do out there, like in the soil, for instance?
That's a great question. In soil ecology, it's actually really important. Oxalic acid, which fungi and plants can secrete, helps make nutrients more available. It can dissolve minerals like apatite.
Apatite, that contains phosphorus, right?
Exactly. So it releases phosphorus and other micronutrients, making them easier for plants to take up. It's part of that whole nutrient cycling system.
And I know you mentioned animals. It has relevance for us humans too, doesn't it? Something about kidney stones.
That's right. Our bodies, well, we don't have the enzymes to break down oxalate effectively, so it mainly gets filtered out by the kidneys.
Right.
If you consume a lot of high oxalate foods, that can contribute to kidney stone formation, a condition called hyperoxaluria. So yeah, it directly impacts human health too.
Okay, so it's everywhere. It cycles nutrients. It can even affect us. How do the microbes themselves actually, make it? Let's maybe start with fungi.
Fungi are big players here, especially ectomycorrhizal fungi. Those are the ones that team up with tree roots. They're known to synthesize and pump out quite a bit of oxalic acid.
Team up how? Like a symbiotic thing.
Yeah, exactly, a beneficial relationship. A good example is Paxillus involutus. It can secrete oxalic acid up to concentrations of like 1 millimole per gram of fungal biomass. That's pretty high.
Wow.
And this secretion isn't random. It's influenced by things like how much calcium or bicarbonate is around. It's their way to dissolve those minerals, get phosphorus, and also, interestingly, lock up excess calcium by forming calcium oxalate crystals.
So it's like biochemical engineering on their part. Very clever.
It is. And there are other examples like Mycena citricolor, which causes a leaf spot disease on coffee, another oxalic acid producer.
Right, I saw that. And then there's the wood-rotting fungi.
Yeah.
They use it defensively.
Yeah, that's another fascinating angle. Some wood rot fungi form calcium oxalate crystals almost like a detox mechanism, especially if they're growing in soil or wood that's contaminated with toxic heavy metals.
So nutrient mining and detoxification. It's a versatile tool for them.
Definitely. It really highlights how fungi use this compound in multiple ways.
Okay, what about bacteria? You said they're involved too.
Absolutely. There's a whole group called oxalotrophic bacteria. The name kind of gives it away. Oxalo for oxalate, trophic for feed.
They eat oxalate.
Pretty much. They can use oxalate as their one and only carbon source to grow. It's their food.
That's amazing. Are they rare or?
No, they're quite diverse and found in lots of places. You've got Specialists like Oxalate flavum, but also common soil bacteria like Streptomyces species can do this. They're crucial for breaking down oxalate in the environment, stopping it from just building up everywhere.
Okay, so some microbes make it, others break it down. Let's talk more about the breakdown crew. What role do they play?
Right, the degraders. This is where things get really interesting, especially thinking about ecosystems and even our own gut. Certain probiotic bacteria, like some bifidobacterium and lactobacillus species, have this ability.
The ones in yogurt and stuff?
Some of them, yeah. They can break down oxalate into simpler things, carbon dioxide and formate. This is thought to be helpful in managing oxalate levels in our digestive system.
How do they actually do it, like chemically?
They have specific tools enzymes, things like oxalate oxidase and oxalate decarboxylase. Think of them as tiny molecular machines designed specifically to process oxalate.
Got it.
And these are coded by specific genes, like the OXY and FRC genes. It's a specialized metabolic pathway.
Okay, so specific bacteria, specific enzymes. Now, zooming back out to soil, there's this thing called the oxalate carbonate pathway. Sounds complex. What's going on there?
It is a bit complex, but super important. It basically describes how breaking down calcium oxidizing it actually changes the local chemistry. It makes the area around it less acidic, more alkaline.
Raising the pH.
Exactly. And that rise in pH can then cause calcium carbonate, basically calcite, like limestone, to precipitate out of solution.
Okay, and how do microbes drive this?
Here's the kicker. It often requires teamwork. Experiments in controlled soil setups, like microcosms, showed something really cool.
Yeah.
They only saw that significant pH shift, say, from pH 5 up to 7.5 over a few months, and the calcite formation if and only if both the oxalate-degrading bacteria and fungi were present together with the calcium oxalate.
Wait, so neither one could do it alone? Not just the bacteria, not just the fungi?
Correct. Neither group by itself was enough to really drive that change. It needed both working together. Synergy.
That's a huge point. Partnership is essential. Did they see this in real soil too, not just sterile lab stuff?
They did. And in non-sterilized soil, with all its native microbes, adding calcium oxalate made the pH rise even faster. Like about 1 pH unit up in just 2 weeks. It hit pH 8 in less than 10 days.
Whoa.
So it reinforces that idea. You really need both the bacteria and the fungi working in concert for this pathway to function efficiently in a complex environment.
This leads perfectly into something I read about called the fungal highway. It sounds amazing. What is that?
It's a fantastic concept, and it's very real. Fungal hyphae, those long thread-like structures fungi grow, can literally act as transport networks for bacteria, especially in places like soil where water isn't everywhere, unsaturated conditions.
Like tiny rows for bacteria.
Exactly like roads. It helps the bacteria move around much better than they could on their own. And crucially, it lets them reach resources that aren't easily dissolved, like solid calcium oxalate crystals.
Ah, so the fungi provide access.
Precisely. It's this incredible physical interaction that allows the bacteria to get to their food source and drive these metabolic processes like the oxalate breakdown we were just talking about. It's cooperation on a micro scale driving macro scale environmental changes.
Hey, this is fascinating. We've got this compound made and broken down by microbes working together. Now let's bring it specifically to mushroom farming. The white button mushroom, Agaricus bisporus. How does calcium oxalate feature there?
It features prominently. Electron microscopy, taking super high magnification pictures, has clearly shown calcium oxalate crystals right there in the mushroom compost and even directly on the Agaricus mycelium, the fungal body itself.
So it's definitely present. Does it show up all the time or at certain stages?
Researchers have tracked it. Around day 35 of the growing cycle, that's typically when the casing layer goes on top of the compost.
Right, the soil-like layer.
Yeah. Around then, you start seeing crystals appearing on the Agaricus mycelium that's growing on the straw bits in the compost.
Okay, so it starts appearing then.
And then by the end of the harvest period, say around day 112, the dense mat of mycelium on the straw can be just completely covered in tons of these crystals.
Really coated.
Yeah, quite profusely. It gives the mycelium this distinct chalky white look. Actually, people noticed this way back. There are descriptions from 1887 of Agaricus campestris, a related mushroom, looking white because of these crystals.
Wow, over a century ago. So they're all over the mature mycelium. What about the growing parts? The tips of the hyphae?
That's an interesting observation from the studies. The crystals were generally not seen right near the growing hyphal tips or on the hyphae that were actively pushing into the straw pieces to digest them.
So maybe it's more associated with older established growth.
That seems to be the implication. Perhaps less metabolically active zones, or maybe it's a byproduct that accumulates over time on the surface?
Okay, so it's there, it accumulates. Does it do anything? Does it affect the process, like maybe with pests?
It seems to. Sciuretted fungus gnats, Leucoriella auricola, are a huge headache for mushroom growers. Their larvae damage the mushrooms.
Right, I know they're a big problem.
Well, some research added fungal byproducts, including mannitol and calcium oxalate, to the compost, and they found it actually slowed down the development of the gnat larva.
Interesting, so it could be the defense mechanism, making the compost less appealing or harmful to the pests?
It certainly points that way. An antagonistic relationship, maybe? The larvae seem to avoid compost that's fully run through with mycelium, and maybe this accumulation of oxalate and other stuff is part of why they avoid it.
Okay, so defense against pests. What about other microbes?
Oh.
Pathogens? Beneficials?
Interactions there too. Crystals have been seen during interactions with Licanicilium fungicola.
That's the dry bubble disease one, right? Nasty stuff.
That's the one. Crystals were visualized when Agaricus was interacting with it, and also after the disease had really taken hold. The significance there isn't fully clear yet, but they are present during infection.
But there's a flip side. A positive interaction involving these crystals.
Yes, and this is really cool. In the casing soil, researchers observed these little rod-shaped bacteria right up close interacting with the Agaricus hyphae. And where these bacteria were present, the calcium oxalate crystals were disappearing.
The bacteria were getting rid of them.
It looked like it. And what's more, this disappearance of the crystals seemed to help the fungus form strands.
Fungal strands, what are those?
They're like bundles or cords of hyphae woven together. They're really important for transporting water and nutrients efficiently through the compost and up to the developing mushrooms. Better strand formation usually means better mushroom development.
So bacteria eating the oxalate actually help the mushroom build its transport network.
That's what the observation suggests, a really positive role for certain bacteria in managing these oxalate levels, potentially boosting the mushroom's growth.
Okay, this paints a really complex picture. The mushroom makes oxalate, maybe for defense, but too much might not be good, and then specific bacteria can clear it away, which seems beneficial. This makes you wonder, right? Could we add these helpful bacteria on purpose?
Exactly. That's the logical next step, isn't it? Given that Agaricus makes a lot of oxalate and we see it building up and we see these interactions, could inoculating with specific oxalate-degrading microbes be a way to improve cultivation?
Manage the oxalate levels deliberately.
Yeah. The thinking is, if we can control that accumulation, maybe we mitigate any negative effects and enhance the positive ones, potentially improving yield or quality.
Have people tried adding beneficial bacteria to mushroom compost before, generally speaking?
Yeah.
Even if not specifically for oxalate?
Oh, yes. There's been quite a bit of research into microbial inoculants for mushrooms. Some studies have shown really promising results. For example, adding strains like Bacillus megaterium or a heat-loving Staphylococcus species to the compost has been shown to boost mushroom production and even make the mushrooms appear a few days earlier.
Earlier harvest is always good for growers.
Definitely. And another one, Pseudomonas purtida, was shown to help the Agaricus hyphae grow longer and spread faster, at least in lab settings. That encourages faster colonization of the compost.
Sounds great. But I guess it's not always a magic bullet. Do these additions always work?
No, not always, and that's important context. Some studies didn't find any significant yield increase when they added other bacteria, like Bacillus subtilis or even Bacillus megaterium in different experiments, along with the mushroom spawn.
Why the difference?
It could be lots of things. The specific strain used matters a lot. The conditions in the compost temperature, moisture, the existing microbes already there. It's a complex ecosystem. Sometimes the added bacteria might not compete well, or the conditions just aren't right for them to have a big impact.
So it's nuanced, it depends.
Very much so. Biology is rarely simple.
Are there other ways bacteria help, maybe indirectly, not just by eating something specific like oxalate?
Yes. There's a neat example with another Pseudomonas purtida strain. This one produces an enzyme called ACC deaminase.
Okay.
What this enzyme does is break down a precursor to ethylene gas. Ethylene, even in small amounts, can actually inhibit the mushroom mycelium from forming primordia, those tiny baby mushrooms.
So the bacteria remove an inhibitor.
Exactly. By lowering the ethylene levels around the hyphae, this bacterium helps stimulate the mushroom to start fruiting. It's an indirect but powerful effect.
Okay, so bringing all these threads together, the mushroom-making oxalate, the pests, the pathogens, the helpful bacteria, the fungal highways, what's the real potential then for specifically adding oxalate-degrading bacteria to commercial mushroom farms?
Well, when you synthesize it all, it looks like a really compelling strategy. We know bacteria are key for breaking down oxalate in nature. We know they often need fungi, like the mushroom mycelium itself, to help them access at those fungal highways.
Right.
And we've seen specific instances in Agaricus cultivation where bacteria do seem to be degrading oxalate crystals, and this appears linked to better fungal structures.
So the logic follows.
The logic follows that intentionally introducing known, effective oxalate-degrading bacterial strains, especially ones that are good at living alongside fungi and maybe even using those highways, seems like a very promising approach.
The goal being to actively manage the oxalate levels.
Precisely. To eventually reduce any negative impacts, maybe enhance pest deterrence in a controlled way, or promote that beneficial strand formation we talked about. Ultimately, the hope is to optimize yield and maybe even mushroom quality.
And it makes a lot of sense. Is this already happening, or is it more of a future prospect?
It's definitely an area of active interest and ongoing research. While we understand the basic microbial interactions pretty well now, we need more large-scale studies directly linking specific oxalate degrader inoculations to consistent, significant yield bumps in real-world commercial mushroom farming. But the potential is certainly there. It's an exciting frontier.
It really is. So wrapping up our deep dive today, calcium oxalate. It's not just some random mineral. It's involved in everything from kidney stones in us to nutrient cycling in forests and intricate microbial ballets in mushroom compost.
Absolutely. Fungi and bacteria aren't just existing side by side. They're interacting, cooperating, sometimes competing, using compounds like oxalate in really complex ways. Those fungal highways are just one amazing example.
And understanding this delicate balance, especially around calcium oxalate in Agaricus cultivation, could genuinely change how we grow mushrooms in the future. It makes you wonder, doesn't it? What other hidden microbial alliances are out there, shaping our world in ways we haven't even realized yet?
A question that keeps microbiologists busy.
Indeed. Well, that's all for this deep dive on the Mycelium Digest. If you found this journey into the world of calcium oxalate insightful, please do hit that like button over on YouTube, subscribe if you haven't already, or perhaps leave us a rating on your podcast platform of choice.
Your support genuinely helps us keep exploring these fascinating, unseen worlds.
Thanks for joining us.